Hey guys, this video is about different signaling pathways. We've got our RasG protein pathway and our Heterotrimeric G protein. So let's go over the RasG first. So if you want to follow along here, we've got this is going to be like our cell membrane, right? So this is outside and this is inside. So we have our little receptor here. That's what that is. So some sort of ligand is going to come and bind to our receptor. And in our receptor, built into this receptor, because it's these are receptor tyrosine kinase. It's an example of receptor tyrosine kinase. So actually, let's write that over here. So this overall is our RasG protein signaling mechanism. So this is a type of receptor tyrosine kinase like example. So on our little receptor here, I'm going to illustrate the tyrosines just with a blue dot, right? So know that this means there's a tyrosine, which is one of the 20 amino acids, so it's just making up part of the structure of that receptor. So what happens when our ligand binds to this receptor? It's going to cause these tyrosines to phosphorylate. So it's all of this activity is within the receptor itself. Remember, kinases phosphorylate things, so the receptor has kinase activity and is able to add little phosphates, which are these P's here, to the tyrosines, and that makes the phosphotyrosine is what they're called now. Phosphotyrosine. So when that happens, we can get our GERB2 is our first guy to the scene. Sorry. Okay, GERB2 is, this is a type of SH2 containing protein. That's important to know. Okay, so GERB2 is going to come, and then our SOS is the next to join the party. So it's going to bind onto GERB2. Our SOS is a guanine exchange factor, which I'll explain in just a second what that means. So SOS, I guess it has the ability within it to be a guanine exchange factor. Which is also, you can abbreviate as a GEF. Okay, and then our RAS is going to join up with all of those. Is that dark enough? So now we have three things all binding together. Once all three are bound, that is going to activate the guanine exchange factor properties of the SOS. So down here, we're going to have a different RAS. It's a totally different RAS. And this RAS currently has a GDP attached to it. So diphosphate, right? So there's two phosphates. So what we're going to do is this guanine exchange factor is going to come in here, the guanine in the GDP. So that's why it affects the RAS. So that is going to come in and how should I do this? So all three of this is one complex. So the guanine exchange factor, we're going to come in here and get rid of GDP completely. We're not phosphorylating it. We're just completely getting rid of GDP, and we're adding on a different GTP molecule. So that is the triphosphate form. So that activates RAS. Okay, so when we activate RAS, RAS can go and activate the next one. So the next guy that gets activated, let me just use that color, is going to be RAF. So RAF is also known as mitogen activated protein kinase. So MAPKKK. So that is just another word, another term that we use for this RAF protein. So we know that kinases phosphorylate things. So this is a kinase that's going to go phosphorylate the next protein, which is MAPKK. So MAPKK is next in line, which is also known as MEK. MAPKK. So MAP kinase. MAP kinase is going to go phosphorylate the next guy, which is ERK, also known as MAPK. So MAP kinase, it has kinase activity, so it's going to go phosphorylate something else. So we have our, let's use red. So we've got our nucleus in the cell. I want this to be bigger, sorry. It's our nucleus. So inside the nucleus, we've got a transcription factor. We don't really need to know what type, it's just a transcription factor. So our ERK, also known as MAPK, is a kinase, right? So it can phosphorylate something. So it was going to come in and phosphorylate this transcription factor. So we add a P, and that causes that transcription factor to become active, and the end product of this whole thing is to stimulate gene transcription. Stimulate gene transcription. So it's really important as we go through all of these different pathways to think about like what is the end goal of each of these, and that'll help you kind of differentiate between them. So the end goal of our RasG protein signaling mechanism is to stimulate gene transcription. Okay, moving on. We're gonna erase this, so if you need it, just pause and keep writing. So we're gonna have to do this one in two parts. So we're gonna do our heterotrimeric G protein next. Can you see that? Yep. Okay, heterotrimeric G protein. No. Okay, so we've got the cell here, right? So this is outside and in. So for heterotrimeric G protein, we have our receptor here. And then we're also going to have our three subunits. So we're going to have beta. Let's draw like 3 circles here for our 3 subunits. And these are kind of like attached to the receptor. They're hanging out there So we've got beta, gamma, and alpha. So we have something like our ligand is going to come and bind to the receptor. So when that happens, our alpha has to dissociate from the triplet here. So for this example, I should have written this first, sorry. We're going to be talking about our phospholipase C specifically. So this alpha subunit that is going to activate phospholipase C, it is alpha Q. Okay, so alpha Q is going to go and activate phospholipase C. So in order to activate phospholipase C, the alpha Q originally has GDP on it, and we have to go in and switch that out for GTP, and that's going to cause it to be activated so that it can go and activate phospholipase C. So phospholipase C is an enzyme, so it's going to go and interact with phosphatidylinositol. It's not writing very dark. What color have I not used? I can use this one. Phosphatidylinositol. Okay, so phospholipase C comes and activates that. So what happens is the phosphatidylinositol is going to split into two different molecules. So it's going to split into IP3 and DAG, D-A-G. So IP3 stands for inositol 1,4,5 triphosphate. He will use IP3 on your exam, most likely. That's what he did on mine, at least. But in case you need to know, it's inositol 1,4,5 triphosphate, so 3P, 3 phosphates, right? And then DAG stands for diacylglycerol. Okay, but that's besides the point. So phosphatidylinositol gets split into IP3 and DAG. And then over here, we're going to draw, just pretend like this is our endoplasmic reticulum. Inside the cell, right? So inside of our ER, inside of this endoplasmic reticulum, we've got calcium stored up. Sorry, it's a 2+, whatever. So calcium. So IP3 is going to come and go to the endoplasmic reticulum. There's going to be like a little receptor here, and it's going to cause calcium to come out of the endoplasmic reticulum. So now calcium is out floating around in the cell. And then we are going to have protein kinase C. Okay, so together, calcium and diacylglycerol DAG, they have to work as a team, and they're going to go activate protein kinase C. And remember, kinases are, they phosphorylate things. So it's going to go and phosphorylate a protein. We don't really know which one. Well, it's going to go phosphorylate a protein. So that's going to cause a cellular response, which is our end result of this pathway. Okay, so to turn off this pathway, we could go up and dephosphorylate this GTP and turn it back into a GDP, and that would turn off our alpha Q. I just realized if you go back to your drawing about that we drew earlier with our RasG protein, remember that second RAS, we replaced a GDP with a GTP to activate it. If you want to turn off that pathway, you would take that GTP and turn it back into a GDP, and that would turn off your RasG protein signaling pathway. Sorry about that. So, cool. So this is our first type of heterotrimeric G protein. So let's erase this, and then we're gonna have one more. We're gonna have our adenylyl cyclase. Okay, so I guess I could have left that up there, but so we're still doing our heterotrimeric G protein. But this is our adenylyl cyclase pathway. Okay, so same thing as before, where we've got our cell, we've got the outside and the in. We've got our receptor here, and we had the beta, gamma, and alpha. So alpha, in this case, is going to be alpha S. So when something binds up to our receptor, that causes alpha S to dissociate. So alpha S is going to come and activate adenylyl cyclase, which is an enzyme. Okay, so we activated adenylyl cyclase. So adenylyl cyclase is going to help us do this next step. We're going to have ATP, and it's going to split up and create ADP. So usually if you split up ATP, you would get ADP and a phosphate, but with adenylyl cyclase and activity, we're going to get ADP and cyclic AMP. So that is our second messenger, right? So this, let me write that down. That's important. So the job of a second messenger is to amplify the signal inside a cell. So we only have one thing that bound to the receptor, but now we're going to get a big end result inside the cell because we're using a second messenger. Right. So down here, we're going to have our protein kinase A. How do I want to do this? Okay, so draw like 4 boxes. So 2 of these boxes are regulatory subunits, and 2 of them are the catalytic subunits. So we've got our regulatory subunit, catalytic subunit, so our cyclic AMP is going to come over here, and it's going to bind. We're just going to draw 2 little circles for our cyclic AMP, binding to our regulatory subunits. And that's going to cause the catalytic subunits to come off and be free. Okay, so when our catalytic subunits have come off and they're free, that creates an active protein kinase A. Right? So kinases phosphorylate things. So we're going to have an enzyme. Doesn't really matter what one right now. So we're going to have an enzyme down here. I'll just draw a little circle. So this is our enzyme. And it's going to have inside of it serine and threonine are 2 amino acids. So we're just going to, I mean, it has a lot of amino acids, but we're specifically going to be working with serine and threonine here in this enzyme's structure. So our active protein kinase A is a kinase, so it's going to come and add a phosphate to those serine and threonine amino acids in the enzyme structure. That causes the enzyme to either turn on or off, depends on what we're doing. So turn enzyme on or off. So our end result is that cellular response. So our end result of both heterotrimeric G proteins were cellular response, and the RasG protein one was to do gene transcription. So something important for you to know is the difference between the AS subunit and the AQ subunit. So the AQ, remember, was with phospholipase C, so think like a Q kind of looks like a backwards P. So AQ goes with phospholipase C, and then the other one is adenylyl cyclase. So for this pathway specifically, if we want to stop it and inhibit it, we have a different alpha subunit, and it's called alpha I, so alpha inhibitory. And it's going to come in and it's going to also work on adenylyl cyclase, but it's going to inhibit adenylyl cyclase. And so the rest of this pathway cannot happen, and we cannot get a cellular response. So remember, with the phospholipase C, we turned it off by turning GTP back to GDP. Same with the RasG protein, but this, we use a separate alpha subunit to turn off the whole pathway. I think that's it. Yep, I think that's it. Let me know if you have any questions, like always, and happy studying.